Welcome to the deep dive. Today we're tackling, well, one of the biggest questions in biology, really, developmental genetics. How does that one single cell, the zygote, actually build us?
Right.
Or, a fly or anything complex?
It's a fascinating puzzle
Hmm.
because fundamentally the genes are all there from the start. It's all about the process, the execution. And our goal today, digging into the source material, is to map out that developmental cascade.
Right, looking at how these, universal genetic instructions get used in just the right way at the right time.
Exactly. And how conserved it is. I mean, think about that mouse eye gene example you mentioned earlier, putting a mouse gene into a fly.
And getting a fly eye to grow on its antenna, it's wild. It just screams that the underlying logic is ancient. But, okay, how do we even get from that single cell to a point where something like an eye gene knows to turn on?
Yeah, good question. We need the basic framework first. So
Yeah.
development, when you look at the cellular level, really unfolds in three key stages. First up is specification.
Specification.
Okay. This is where a cell gets its first sort of molecular hints about its identity, like a general neighborhood assignment. You're destined for somewhere up front.
Got it. Like being told you're going to be in the head region, generally speaking.
Precisely. Then, the next stage is determination. This is really the crucial point. It's when that cell's fate becomes locked in, irreversible.
A point of no return.
Exactly. Even if you, say, move that cell somewhere else in the embryo, it would still try to become what it was originally determined to be.
Okay, so it's committed at the final step.
That's differentiation. This is where the cell actually becomes its final specialized self. It takes on its adult form and function. Like the example in the book, a pancreatic cell starting to churn out insulin.
Well, and the engine driving all this,
Yeah.
it's not that cells are chucking out genes they don't need, right?
No, not at all. That's the cool idea.
Yeah.
The variable gene activity hypothesis. Every single cell, well, pretty much every cell, has the complete genome, the whole library.
So differentiation is all about which books you open, which genes you turn on.
That's it exactly. Differential gene expression, only activating the specific subset of genes needed for that cell type, for that job.
Okay, but if a cell is determined to be, say, a neuron, how does it stay a neuron? What stops it from, I don't know, deciding to be a muscle cell later?
That's where epigenetics comes in. It's absolutely fundamental.
Right, the layer of control on top of the DNA sequence itself.
Precisely. Differentiation and keeping that cell fate stable relies heavily on epigenetic regulation. We're talking about things like DNA methylation.
Adding little chemical tags to the DNA.
Yep. And modifications to histone tells the protein's DNA wraps around, plus the role of non-coding RNAs. All these things work together to shape the chromatin structure.
And that structure basically dictates which genes are accessible, which ones can be read.
You got it. It creates these stable patterns of gene activity. It physically locks in that cell's identity, making it heritable through cell division.
And this whole process starts incredibly early, doesn't it? The source material mentions this big wipe clean moment.
Yeah, the global DNA demethylation right after fertilization. It basically erases the epigenetic marks inherited from the sperm and egg.
A fresh start.
A fresh start. But then, almost immediately, new methylation patterns start being laid down, and these new patterns are linked directly to those first steps, specification and determination. It's how the potential of cells gets gradually restricted.
Which explains the difference between, say, totipotent cells?
Right, the zygote itself, or the very first few cells. They can become anything, the embryo proper, the placenta, everything.
Versus pluripotent cells later on, like embryonic stem cells.
Exactly. Pluripotent cells can still make any of the, 200-odd adult cell types, but they can't make the placenta anymore. Their potential has been narrowed. Epigenetics helps lock in those limits.
This incredible conservation of mechanisms across species, that's the heart of Evo-Devo, right? Evolutionary developmental biology.
Absolutely. It's the realization that despite the huge differences between, say, a zebrafish and a zebra, the underlying developmental control genes and signaling pathways are remarkably similar. Shared toolkits.
Which is why studying fruit flies or worms can tell us so much about our own development.
It's indispensable. And for a master class in this kind of sequential hierarchical development, Drosophila, the fruit fly, is just perfect.
Especially those really early stages. It's not like typical cell division at first, is it?
No, it's quite unique. After fertilization, the nucleus divides many, many times. But the cell itself doesn't divide. You get this multinucleated cell called a syncytium.
So hundreds of nuclei just floating in a shared cytoplasm.
That's right. Then they migrate to the outer edge, and then cell membranes form around them, creating what's called the cellular blastoderm. It's a very efficient way to quickly set up a field of cells.
And laying out the body plan depends on two sets of genes working one after the other.
Correct. First, you have the maternal-effect genes. Crucially, these are genes from the mother. She transcribes them, and the mRNA or proteins are loaded into the egg cytoplasm before it's even fertilized.
So the egg comes preloaded with instructions from mom.
Exactly. Often in the form of gradients, like the famous bicoid protein gradient, high at one end, low at the other. That gradient basically tells the embryo, this end is the head, that end is the tail. It sets the main anterior-posterior axis.
Okay, so mom sets the initial broad coordinates. What then?
Then those maternal factors activate the second set, the zygotic genes. These are the embryo's own genes, transcribed from its own nuclei after fertilization.
And these genes read the maternal cues and start building the actual segments.
Yes, this is the segmentation cascade. It's this beautiful hierarchical process that won a Nobel Prize. It happens incredibly fast, too.
Step one of the zygotic genes.
The gap genes. They're switched on by the maternal gradients. Think of genes like hunchback, cripple, NERPs.
And gap because?
Because if you mutate one, you get a big gap in the body plan, like missing the whole thoracic region, for instance. They define the broad territories: head, thorax, abdomen.
Okay, broad regions established.
Then the gap genes activate the next layer, the pair rule genes. Names like even skipped or Fushitarazu. These genes divide the embryo into a series of stripes, usually about 2 segments wide. They establish the basic periodicity.
So if one of these is mutated, you might see effects in every other segment.
That's typically what happens, yeah. You get this repeating pattern of defects.
And the final refinement.
That comes from the segment polarity genes like engrailed or wingless. They're activated by the pair rule genes. Their job is to divide each of those broader stripes into 2 distinct segments, defining an anterior and a posterior compartment within each one.
So that gives you the final count of, what, 14 distinct segments?
Exactly. It's like focusing a lens. Maternal gradients give the wide view, gap genes block out regions, pair rule genes draw stripes, and segment polarity genes define the final detailed segment boundaries.
Okay, the segments are marked out, but a segment in the head needs to be different from a segment in the thorax. How does each segment know what it's supposed to become?
Now we get to the identity crisis. That's the job of the homeotic selector genes, often called Hox genes. They step in once the segments are established.
And these are the ones that lead to those really dramatic mutations, right? Like the fly with legs on its head.
That's the classic example, the antennapedia ant mutation. It's a gain-of-function mutation where the antome gene, which normally specifies leg identity in the thorax, gets wrongly switched on in the head segment.
So instead of making an antenna,
it makes a leg, growing right out of the fly's head. It's a startlingly clear demonstration of how a single gene can act like a master switch, dictating the identity of an entire body part.
a developmental switch. And these Hox genes themselves, they share some common features.
Oh, absolutely. They all contain this highly conserved DNA sequence, about 180 base pairs long, called the homeobox.
The homeobox.
Right. And the homeobox codes for a 60 amino acid protein domain called the homeodomain. This homeodomain is what allows the Hox protein to bind to DNA. It's a DNA binding motif.
So they act as transcription factors controlling other genes downstream.
Exactly. They regulate batteries of target genes to implement the specific developmental program for that segment, build a wing here, build a leg there.
And the way they're arranged on the chromosome is also significant. Something about colinearity.
Yes, colinearity is fascinating. The order of the Hox genes along the chromosome actually mirrors the order in which they are expressed along the anterior to posterior axis of the embryo. The first gene in the cluster patterns the head, the next one patterns the next segment back, and so on.
Wow. So their physical location dictates their spatial expression. And this isn't just flies, right? This holds true for us, too.
It absolutely does. Vertebrates, including humans, have 4 clusters of Hox genes, HOXA through HOXD, showing the same colinearity principle, and they're crucial. Think about the run gene homolog in humans, RUNX2.
What does that do?
It's vital for bone formation. Mutations in RUNX2 lead to a condition called cleidocranial dysplasia, or CCD.
And that causes
skeletal defects. People with CCD often have underdeveloped or absent collarbones, dental abnormalities, and sometimes delayed closure of the skull bones. It really highlights this deep, conserved role in fundamental processes like building the skeleton.
Okay, so this Hox system is ancient and powerful in animals. Did plants, when they evolved complex body plans like flowers, just borrow the same toolkit?
You might think so, but no. It's a fantastic example of convergent or maybe parallel evolution. Plants absolutely have homeotic genes that control organ identity, like in flower development.
Flowers having different parts like sepals, petals, stamens, carpels.
Right, arranged in concentric whorls. And specific genes determine which whorl develops which organ type.
Yeah.
The genes doing the job in plants, like in the model Arabidopsis, belong to the MADS box family of transcription factors.
MADS box, not homeobox.
Completely different protein structure, no homology to the animal homeobox domain. So plants evolved their own independent genetic system to solve the same problem. How to assign identity to different parts of the body plan. Same principle, different toolkit.
That's amazing. Okay, shifting gears a bit. We've talked about gradients and transcription factors setting up patterns, but development also relies heavily on cells communicating directly with their neighbors, right? Influencing each other's fate choices.
Oh, absolutely critical. And a great system to study that is the nematode worm, C. elegans. It's famous because it has a completely fixed number of somatic cells, exactly 959, and their entire lineage, who divides to become whom, is known.
So you can track individual cell decisions. What's a key signaling pathway here?
The notch signaling pathway is a major one, and it's all about direct cell-to-cell contact.
No signals drifting across distances, they have to touch.
They have to touch. One cell presents a signal protein on its surface in C. elegans, a caught in what is called delta, or LAG2 in certain contexts. The neighboring cell has the receptor protein, notch, or LIN12.
Delta meets notch. What happens then?
When they bind, it triggers an enzyme to cleave off the intercellular part of the notch receptor, the notch intercellular domain, or NICD.
Oh, as piece breaks off inside the receiving cell.
And that piece, the NICD, travels directly to the nucleus. It doesn't need a complex cascade. It goes right to the nucleus and acts as part of a transcription factor complex to change gene expression in that receiving cell.
Wow, that's very direct. Touch leads to nuclear action.
Extremely elegant. And you see it play out beautifully in things like the decision for cells in the developing vulva. There are two initially equivalent cells, Z1.PPP and Z4.AA. They basically compete.
Compete how?
Through notch signaling. By chance, one might start expressing slightly more delta signal. This stimulates notch activity in its neighbor. The neighbor, receiving the stronger notch signal, is induced to become the uterine precursor cell, VEU. The cell sending the stronger delta signal adopts the alternative fate, becoming the anchor cell, AC.
So it's like a feedback loop where a small initial difference gets amplified by this direct signaling, forcing them into different fates.
Exactly. It's a lateral inhibition or competitive mechanism mediated by notch.
So we have gradients, segmentation genes, identity genes like Hox, and now cell signaling like Notch. All these layers feed into the big decisions. Which brings us back to those master regulators, the binary switch genes.
Right. These are genes that seem to sit at the top of a hierarchy for developing a whole complex structure, like an eye. They initiate the entire program.
And the classic example is the eyeless gene in Drosophila.
The very same one. If a fly has a loss-of-function mutation in eyeless, it lacks eyes. But the really stunning experiments were the gain-of-function one.
Where they turned eyeless on in the wrong place.
Exactly. If you force the eyeless gene to be expressed in, say, cells that would normally form a leg or an antenna,
you get an eye growing there instead.
you get an ectopic eye, a structurally recognizable, although maybe not perfectly functional, compound eye growing right out of the leg or the wing or antenna. It proves eyeless is sufficient to trigger the eye development pathway.
Incredible. And the punchline, connecting back to our very start, is the conservation.
The conservation is astounding. The vertebrate equivalent of eyeless is a gene called PAX6. They share significant sequence homology, particularly in their DNA-binding domains.
So similar genes doing the same job in flies and mice, despite eyes looking totally different.
And the killer experiment was taking the mouse PAX6 gene and putting it into a fly.
And did it work? Could the mouse gene train fly eyes?
It worked perfectly. Expressing mouse PAX6 ectopically in the fly caused the formation of ectopic fly eyes. Not mouse eyes, fly eyes.
Whoa! So the switch gene is conserved, and the downstream network it activates is also conserved to the point where the mouse switch can operate the fly machinery.
That's the interpretation. It tells us the fundamental genetic logic for build an eye is ancient, shared between insects and vertebrates, even though the final structures diverge massively over hundreds of millions of years.
So wrapping this up, the picture that emerges from this deep dive into the source material is one of intricate hierarchical control.
Definitely. It starts broad with maternal gradients, gets refined by segmentation genes, given identity by homeotic selectors like Hox genes, all driven by differential gene expression.
which is locked in by epigenetics.
and fine-tuned by cell-to-cell communication pathways like Notch. It's a cascade.
And the power residing in some of these key regulatory genes, these switches, is immense. Which leaves us with a pretty provocative thought for you, the listener.
Yeah, think about this. The source material touches on regeneration, mentioning work in planarians, flatworms. Some species can't regenerate their heads, but researchers found that by inhibiting just one signaling pathway, the 1-to-beta-catenin pathway, they could induce complete head regeneration in these deficient worms.
Just flipping one switch enables a whole complex process.
So given how conserved this developmental toolkit is across the animal kingdom, what does that imply? If silencing a single pathway can restore such a complex ability, what potential lies hidden within our own genomes, perhaps controllable by manipulating these ancient developmental switches for regeneration or repair?
Something to think about.